Hi, everyone. I am Susanna. I am one half of the team of Fresh Press Science, Nexus Scicommunity YouTube channel.
I'm in Boston. I'm Gabi, and I am a grad student at MIT.
I am a grad student down here in Chapel Hill. Both Gabi and I are trained scientists and science communicators, and we realize, though, that anything that's outside of our field.
I'm a geologist. I study past climate change.
And I could talk until I pass out about microbiology, but the moment I get into a different field, that's not always the case. Each week we dig into a new topic that's making headlines in the media. This week we're going to be talking about the topic of biohacking. Had you ever really heard of this?
The guy who injected himself with something on stage, and it was this big drama thing. Oh, the other thing I heard of is injecting some kind of a chip into yourself, and then you can be RFID scanned.
I feel like I would just call that I lose my stuff all the time. I keep thinking of it as X-Men, where they have some sort of genetics.
I think so too. Yeah, that's kind of the more mainstream or dominant narrative that ends up happening.
Is biohacking really a thing? Are people actually doing it? You see media stuff, but are people just kind of in their garages? Does it translate at all to what people are doing in the lab? I think it came out of this kind of citizen science. It's something that is not regulated by bodies.
But it's really important to have bodily autonomy and to have your body behave the way that you would like it to be. But I think it gets really dangerous when people are injecting yourself, for example, with E. coli because it would turn you green. I just made that up. I would not do that.
It's getting the same kind of coverage and the same narrative, the same story as peer-reviewed, really rigorous experimentation. So I guess that's my concern.
I'm just still thinking about this paper that we read to talk a little bit about this, but I'm frustrated because I understood so little of it. How is biohacking happening now in the lab? Or what is the real science behind biohacking, if there is any at all?
I think that's actually the big question, is that in the lab you have to test everything. You have to, or at least you should, understand where all these things are coming from. And in your basement or whatever, maybe not the case, but what's really happening is all this intricate science. So they also might not know these big terms that would give them more knowledge about exactly what they're doing. It's sort of this random chance versus what we would think of as science in a lab.
Yeah, it would help to be able to make an informed decision about whatever you want to do. And so if you can read scientific research papers about protein engineering and changing your DNA or organism's DNA to produce what you want, then maybe that would help you make a good decision about it. But it can be difficult to get through those papers as someone out of the field.
Oliver, we love you. This is horrific. It's so well done.
No offense to the paper, right?
No, the paper is great, but I can't read this.
I should have taken some kind of, I don't even know what class I should have taken to be able to understand this, I have to say. Check out the chat because Oliver put the link in the DOI right there. Italicized TRP. TRP trip. RNA binding attenuation protein, modifying symmetry and stability of a circular oligomer. That's going to be my personal brand of this entire show, is me trying to pronounce things and putting the emphasis in the wrong place. Okay, so circular oligomers.
I guess we should start with breaking down the title. Yeah, circular. So circular circles, and you saw that in the picture that Gabi just put up. It's awesome. It's circular. We'll have to ask him why exactly that's important. I think people get really jazzed about circular proteins because it doesn't, how does that circularize? Oligomer. So are you familiar with the word oligomer?
Only because Oliver wrote a nice explanation here that I kind of understood. So he was saying that it depends also if you're a chemist or a biologist, where I think he said that if you're a chemist, it's more small scale. If you're a biologist, you're linking proteins together.
Oligomer, right? So oli meaning many. So polygon is many. An oligon would be just it tells you that there's multiple subunits in that. So
an oligarchy, maybe people are familiar with.
There's multiple individual pieces of it. What Oliver was trying to do, could you guys chill? Really? What Oliver was trying to do in this paper, as I understand it, is that They have this protein called TRAP. They think if there is a different number of subunits, there would be more stability. And so stability in this case means that the enzyme, this protein, would keep working for longer, maybe at higher temperatures or things like that. I know at least in biology, a lot of times you want to be able to use enzymes and chemicals at a higher temperature, and that way things can go faster. Or sometimes bioreactors just get hotter. So we'll have to figure out exactly why he wanted that. We're going to learn how to make a super protein. We're going to learn how to make a super enzyme. And we're going to start with this circular protein, and hopefully it's just really fast and easy, right? Just like stick it in a tube, get done with it. You made a super protein, move on.
Like a super protein in that it could live in this high heat environment, for example.
So what makes enzymes special versus something else is that they don't run out. For as long as they're stable, as long as they're existing, they're going to keep doing the same thing. So like putting somebody into a room and saying, make this box every single time. So anytime you get this piece of paper, make this box. And then once the protein unfolds, once it loses its stability, it can no longer do that function. And so the idea is that basically using this analogy, you would, instead of having a worker that could go in and do box making for a few hours, you now could have it go in and do it for a few days. Cool.
But I guess the concept of biohacking is like, is it true or is it plausible that someday we could have the ability to like make all of our cells and all of our enzymes resistant to high heat? And then we could have like genetically engineered firefighters who can go save people from burning buildings without getting burnt.
Yeah, we should totally ask about like how this would affect cell function. Like, would this affect the bacteria?
Because that blows my mind. I'm afraid of it.
It's so pretty. Almost all of this, to me at least, seems like characterization. And there is so much characterization. Like, I would think that if you wanted to make a protein better, you would have maybe one metric that you were measuring on. But it looks like they've got a whole bunch of stuff. They've got a bunch of like cartoony looking figures with arrows. It's assumed that people who are reading these types of papers are experts, which is totally makes sense. Like if every single scientific paper had to be broken down into its tiny pieces, they would be a book long each. That's why we have textbooks. Can't expect the regular layperson to just be able to jump in and read any scientific paper.
I just wish I could make cool like mesh looking figure things like that. You see that? Why does it say 11-mer and a 12-mer? Is that how many subunits it has?
If it had 6, it would be a hexamer or a 6-mer.
Oh.
It's like, as soon as I get out of my field, I'm like, have I ever learned anything else in my track of, are you talking about the protein that has 11 or 12 subunits?
To me, like a protein nightmare. I should stop being so negative about this, but I read like one sentence of this, and I think I texted you immediately and was like, I can't do this. Like, what the heck is this paper?
Hello!
Hey!
So where are you?
I am in Norwich, which is like in the south of England. It is like 11:30, about that. So I'm apologies if I come across as being a bit sleepy.
It's like 6:30 here, and I'm ready for bed.
We have questions for you, Oli.
Okay, yeah, shoot. Yeah, I feel like I have to apologize for the paper because there's just like so much stuff in it, and you could just like watching you two talk about it makes me realize like how really like specialist knowledge you have to understand it, whereas we just pick it up and go, oh yeah, like, whereas like, yeah, I see now that it's pretty awful.
No, it's not awful, it's really
no,
cool.
but you know, like accessible, like to the,
as scientists, maybe we should be the ones reaching out to the media, or you know, I like that some papers now have you do something where you actually try to translate it to a general audience. So what's our donut? What's the donut thing?
So the donut, so the picture is like we call it a ribbon diagram. It's like a particular way we visualize protein structures, like atomic structures of a protein. And that protein is called TRAP, or tryptophan RNA binding attenuation protein. The trip, like you said, why is it in italics? So it's in italics because it denotes that it's the gene that's involved in like tryptophan biosynthesis.
this is my question, because
Yep.
I know that, but it's capitalized. Is that just a paper thing?
First
Yeah.
word, is it? Yeah, probably just because it's the first.
It's a title thing.
I feel like we're like critiquing your paper, and we're not at all.
Well, that's what it's for. Yeah, no. Gritty donut. Yep. Yeah, so it's trap protein. It's colored by, I think, subunit. So we color it round. Like one subunit will have a different color. It just enables you to see to pick out individual like protein monomers. There's a little like atomic blob in each subunit, like little tiny blobs stuck together. Do you see that?
Yeah.
Like they're tryptophan molecules. So each one is a tryptophan amino acid, just one molecule bound in like a central pocket.
Oh.
So that is the main structure that's reported in the paper. And because we're in a structural biology lab, like most things the story tends to be based around a structure or two or three structures.
What is structural biology? Like we've been talking about biohacking.
We're determining the structures, like the 3D structures at the atomic level of protein, mainly proteins or nucleic acids or any biological molecule like carbohydrates, lipids, sometimes using either microscopic techniques or like diffraction techniques. So that's probably quite specialist knowledge what those are as well, actually. So like either using electron microscopy or X-ray crystallography, where you like shoot your protein with X-rays and you do a load of maths to reconstruct the 3D blob.
I love that description, because I feel like a lot of times when scientists are trying to describe their own research, they still have to talk at such a high level. But I mean, that is what X-ray crystallography is. Figure out the blob shape.
Yeah, the blobs. It's all blobs, really.
Kind of related to that, someone just asked in the chat how the crystal structure looks. So I think, and then it says how the donuts like to stay next to each other. So I guess how did the donuts fit in? If you had multiple traps next to each other, how would they interact?
Or maybe they've mutated them so they form like these long nanotubes, like these long wires. So if you got tons of donuts stacked on top of each other, the theory being you could somehow use that to conduct electricity, maybe if you could decorate it with metal. But I don't think they do that normally in their like native role. That would be sort of like the protein engineering, like using a protein for something that it's not evolved to do.
Did you hear about my crazy idea to make a super firefighter?
I did. That would be cool. I was trying to think whether it'd be possible, because like I work with a lot of like thermostable bacteria and bacteriophages that infect Thermus thermophilus, so they grow at like 70 degrees or 80.
So like, could you make a suit, maybe like a bacterial biofilm suit that could
just like
protect
form
you?
around you.
It used to be disgusting. It's like a slime monster.
Okay, that's what you do all day, Susanna. You look at slime. So
But I
what's the
don't
difference
cover
between
myself.
that and putting it on myself?
Now I really wonder what you think I do in lab. Like I just go in and just like smear bacteria on myself. You were trying to change stability, things like that. Why?
Why? What are you doing? So, well, the objective wasn't really to change the stability. That was just like one of the measures we took from it. So the idea was just to understand why you have 12 subunits in a ring, basically, or why you have 11, and that varies from one species to another. And you can do that like in vitro if you like snip a bit off the end of one of the subunits, and it suddenly forms a 12-mer when it normally forms an 11-mer, or vice versa. And you're like, oh, this small bit of the protein is important in determining its shape. And then you might want to look at its thermal stability and say, oh, it's actually more stable now as well, or oh, it's falling apart at room temperature, which means you've.
And when you say you like snip a little piece off, can you tell me how that happens? Like, how do you do that? Is that like weeks of work, or is that literally just like?
It's probably like about a week's work, probably,
Okay.
at the genetic level. This is sort of the biohacking that you were talking about. So you would mutate your gene. You'd have like your wild type gene, and you'd mutate it using something called PCR. I don't know if you're familiar with that, but you use you mutate it in a tube, and then you put it into a bigger piece of DNA, and you give that to the bacteria, and it will produce that mutant protein.
A plasmid.
A plasmid. Yeah.
Really?
It is plasmid, yeah.
That is the level at which some biohacking is going on, is this idea of introducing new or different genes into bacteria. Could you have done this work in your basement or garage?
I think now you probably could. Oh, I mean, even probably like 10 years ago, you probably could, because we're using techniques like PCR and stuff that have been around for quite a while now. So in bacteria, maybe eukaryotic cells and other systems would be more difficult to work with.
What reaction does this thing do?
It binds tryptophan.
Yeah, it binds tryptophan. So it's involved in a regulatory protein. So bacteria need to produce like their own tryptophan to make things, proteins. They need to know how much tryptophan is around. So if they bind tryptophan and they recognize this sequence which is involved in making tryptophan, then it's like the sensing mechanism.
I wrote turkey on my printout of the paper. That's about as far as I got. But isn't that fake news? Doesn't turkey not even actually have tryptophan? Or does it just not actually make you tired?
That a lot of other food has more tryptophan in it.
Ooh.
So it's probably also all of the other stuff you're ingesting along with it. But tryptophan is just one of the amino acids, right? Okay. Oh, yeah, we had questions about this like pretty mesh structure.
Figure 2. In this figure, there's like two subunits. In A, that is, there's two subunits side by side, and they're comparing two mutants. So like, I think one's the wild type and one's the mutant, or at least two variants. And on the right, the mesh is the electron density that's present in one map, one structure, but isn't present in the other structure. And that's just like a structural biologist way of showing that this region is definitely not there. It's like a difference map, we call it. So it shows that we've deleted this region at the genetic level, and when we've solved the structure of this protein that we've purified, it's definitely gone. So it's just like a confirmation of that.
Cool. And would these kind of experiments be possible in your own house? Like, it sounds like changing things is possible, but would it be possible to verify all of this and like make sure that you hadn't introduced random mutations?
Probably like at the genetic and protein level you could, but like it's going to be difficult to like set up an X-ray generator or an electron microscope in your basement probably. So the structural biology might be a bit harder. Yeah, you could do assays and maybe sequencing depending on.
How does changing the trap, how does it change cellular structure? Sorry, my cat almost just fell on me. How does it change the cellular behavior? Like, if you put a bunch of this mutant trap stuff, what does it do?
That's probably like, because like in one of the questions we were asking before, like what would you do as a follow-up? That's probably something that we haven't really looked at, or wasn't looked at in this study. Like this was very sort of in vitro, taken outside of the native
Not in
system.
the cell.
Yeah, so we don't really know whether these are functional or what they do in vivo.
So it's almost kind of outside your job description to figure out if I put a pile of this into a cell, will I kill it?
I mean, you probably would do that as part of a bigger study, but like to verify your like model that you're putting forward probably. But I guess with structural biology, as the name implies, is on the structure and then in vivo stuff.
In this case, you're meaning inside of a cell, right?
Yeah, inside of a bacterium, rather than just like one protein inside of a test tube. Yeah.
So one of the things that got brought up earlier is like there's a difference between if you're a biologist versus a chemist and what an oligomer is. But also I like the words like in vitro and in vivo. And I read a paper today and they were talking about ex vivo. People who study diseases might consider a live bacteria on its own to be in vitro. I think there's this general misconception that scientists all have this secret language, but really we like all have our own secret language.
Yeah, no, that's true. Yeah, I noticed that recently as well, like people talking about ex vivo, and it's like, but that's in vivo, I think you'll find. What
happens when you open the cyclic structure, as in make it linear?
Like to make it like instead of a loop, like if you broke it?
Yeah, what if you just like...
I don't know how you'd actually induce that, like in a controlled way, because you'd probably just make it all fall apart, or...
How does it circularize?
So once each chain has been like synthesized and it's folded into its little monomeric subunit, it will just eventually come into contact with another one of its partners and interact with it. We or hydrogen bonds or like non-covalent interactions, which is again quite like specialist language probably, but they bind to each other, and then they do that in a way that gradually builds a circle. So it's kind of a random process.
Are similar cyclic protein oligomers present in different bacteria? Is it possible to manipulate them as a way to improve antibacterial treatments?
Are other proteins in the cells similar to this, and could this be an interesting like drug target? Are the things that you are learning from this study going to be useful in human-centric applications, or is this kind of more of a pure study?
That's a good question.
So I mean, there's going to be lots of different types of oligomers which form like circles or closed loops in bacteria and in other species like us. And I suppose he's asking like whether the findings of this paper, like manipulating the oligomeric state, can impact on drug resistance. Is that?
Could you have a special antibiotic that fits into the donut and like on the cell?
That would be
I'm
cool.
picturing.
You might be able, like, maybe if
With protein oligomer that could induce a ring opening reaction. On this donut, are there places where you could cut the donut?
when I was talking about snipping earlier, I was talking about like genetically truncating the construct, whereas snipping, I guess we could mean like proteolytically cleaving with an enzyme. Ooh, that's a tough question. I'm trying to think if there are sites on the cyclic protein that could induce a ring opening reaction, like specific. So what we were doing sort of was like introducing bulkier groups that sort of prise open the subunit interface to like change the angle, which changed the oligomeric state. So maybe you could try and do that with a drug somehow, or maybe a protein, but that's quite speculative.
Regarding in vitro applications, in the intro you speak about DNA translocating devices. Is this protein a good candidate for that? And can you give an overview and compare it with inorganic nanopores?
So when we make the reference to like these viral translocating motors, that's quite a leap that probably not a lot of labs would do. It's just because in our lab we work with a lot of viral motor proteins. So like viruses use these little molecular motors to package their genomes. Some of our research is like looking at ways we can stabilize different complexes, viral complexes, using proteins such as this trap protein. So just using it as like a scaffold, even though it doesn't have anything to do with its function, we can maybe just use it as a building block sort of to help something else assemble.
Well, I just asked the audience what's a nanopore.
What was the last question about nano something? It's like
comparing it to inorganic nanopores. What the heck's a nanopore? I mean, I understand the concept of a pore that's very...
If you have like a thin layer, it can be a solid metal, or it can be a protein or a lipid layer. If you have like a little hole in it, we call it a nanopore, and you can have like another circular protein like this one that would make a hole that would like and then you could like push stuff through it. And these techniques are used for like, we're trying to be developed for like DNA sequencing and stuff like that. I don't know if this protein trap has been used in that application, but other sort of circular proteins have been used for like that sort of thing.
So the other link we included, I think, in the stuff was an article from MIT News, so that you know that it's really legitimate because it's from MIT.
Do you think I'm legit because I'm from MIT?
I know you're legit. I know that MIT is legit because of you.
Oh, that's so touching.
They're so like innovation focused, and I think this really talks to that, which is inexpensive biology kits offer hands-on experience with DNA, and then they have a small child playing with PCR tubes, talking about like protein engineering classrooms. These scientists have come up with the ability to freeze dry enzymes. They say molecular machinery needed to translate DNA into proteins, and then they give them a little solutions and they mix it together, and they actually can produce their own proteins. And they relate it to a chemistry kit, which I think a lot of, I don't know, I had a chemistry kit when I was growing up. Did either of you have?
Yeah.
I just realized this is my friend's paper. This is like an acquaintance of mine's work. I like just put together that's the same person, but they're currently trying to find funding to actually make it happen. So it's a pretty cool kind of mission, and it would be cool to think of that happening in the future.
They said like it's a $15 device that could actually read fluorescent. We don't need to get into necessarily what fluorescence is, but when you hear about things glowing green, pigs that have glowing noses, they're expressing this fluorescent protein.
GFP, green fluorescent protein. I put that in a plasmid one time in biology. Susanna's just like, oh my gosh, we're never doing a biology paper again.
Oliver, how do you feel about biohacking? Is it something you've ever looked into?
As in biohacking humans or just animals? And is that what you mean? Or
Sure.
sure. So one thing I was reading about recently, because they've been doing like, have you heard about CRISPR? Yeah, so like this new gene editing technology. In some countries, maybe where restrictions are a bit less, like maybe in China, they've produced like these really muscly dogs by knocking out this protein involved in skeletal muscle growth. I don't know if you saw that. It's probably in the news. This protein called myostatin. Yeah, and so like they think maybe that could be useful in treating Duchenne's muscular dystrophy or something, like these muscle wasting diseases. So I think in those sorts of contexts it would be potentially beneficial, but it usually gets quite bad press, I guess, gene editing and like interfering with what's natural. Yes.
But Gabi, would you ever start biohacking?
Absolutely not. I think you guys are crazy. But also my lab has like a small amount of radioactive stuff, and there are people who would like absolutely not set foot in a lab with that. So I think it's just about what kind of risks you're okay with taking and the safeguards and all that.
I suppose it also depends on like how you define biohacking, like because we do a lot everyday science and like biology where you wouldn't, it's not sort of in the same sense as the Incredible Hulk sort of thing, which is how it's like often portrayed in media.
Yeah, Hulk dog.
Hulk dog.
So my summary, what have I learned? Wow, I've relearned that I don't want to do structural biology because like what is hiding behind all of these beautiful pictures is a whole lot of math, which is not my favorite. I also learned that the exact number of the same subunits could change the property of proteins, which I really don't think about often.
I learned a lot of vocab. I learned that I don't want to be a biologist. And I have new appreciation for your research, Oli, and the cool stuff that you do. So now when I see your Instagram post, I'm going to be like, that guy's a genius. Like,
were there a lot of the maths and stuff like that? Like a lot of it is like computation. A lot of it is just working with proteins and moving small volumes of liquid from one tube to another, like most sciences. Yeah, I guess I've learned that like even though we're writing these papers and we think they're still accessible to like the we could say like the specialist, non-specialist or something like that, and like the public might struggle a bit. I think if you're slightly outside of this field, a lot of people would struggle and the public probably wouldn't have a chance.
What about biohacking houseplants? So glow-in-the-dark roses, like flowers that wouldn't go bad so fast, then maybe I could actually justify buying them.
I would totally biohack my houseplants. I'm really into my cucumber plant and my dill and tomato plants and all those. So if I had a chance to like make them even cooler than they already are, I would 100% do that.
This was awesome. I think we'll let you get to sleep because you've been
traveling. Yeah, thank you.
Yeah, no, thank you for inviting me. It's been fun.
For anyone who just joined at the end, sorry, it's over. But this was Fresh Press Science with Susanna and Gabi, where we talk about science that we have nothing to do with in our real lives, research lives. And there's a bunch more episodes, so you can watch those.
If you're an author on a paper from like the last 5 years and you want to come on here and let us ask really silly questions about your science, we would love to talk to you. So please send us a message. You can find us on Twitter at Fresh Press Sci. You can also send messages through the Scicommunity, both on Twitter and on Instagram. And then my handle is Susanna L. Harris.
Mine is G. Serrato Marks. They're linked in the YouTube description, so you can click on us.
And Oli, where could people, I mean, are you interested in letting people ask you questions? Or are you like, I'm done with questions forever?
No, sure. Like more questions would be fun. So you can find me on Instagram mainly at so-called expert, where I hang out most. So that's probably where you can find me.
We're going to go and think more about biohacking. So this was really fun. Thank you.
Thank you so much, Oli. Thank you for watching.
Bye.
Bye.